Good day, and thank you for attending your lecture. For our meeting today, we will tackle your module 3, which will deal with cell migration and adhesion. So during the discussion class, if you want to follow along, please open your module packets. They are found in your NEO courses. So that you can use them as a guide as the discussion is ongoing. So let's begin. So for this module class, we're going to tackle your cell migration, which will deal with leukocyte migration. Here you will understand how leukocytes would migrate to the site of injury or infection. Then two, the migration of T cells and the migration of B cells. So we will tackle them one by one. And the second topic, we will deal with your inflammation. What is acute inflammation and chronic inflammation? Now let's start first class with your leukocyte migration. Now, whenever we will deal with cellular migration class, we would often involve this with your WBC, specifically the commonly one that would react whenever there would be a site of injury. An example of that class is your neutrophil, your monocytes, and the different lymphocytes, such as your T cell and your B cell. Now, cellular migration class has three functions. The first one is the delivery of leukocytes of the myeloid lineage. Now, when we deal with your myeloid lineage, this would mainly include your neutrophils and your monocytes. So they be delivered from the circulation and then into the tissue sites of infection or injury. Now, this neutrophil class and monocytes are typically found in your peripheral blood. When you say peripheral blood, we're talking about your blood vessels or your circulating blood vessels or the circulation. So the neutrophil and the monocyte would just continuously circulate along with blood. Now, one of the functions here is that from the circulation class, whenever there would be a infection or injury, they would travel from the circulation to specific tissue sites or infection or injury. So we're going to tackle how that is done for this module. Then delivery of lymphocytes from their site of maturation class, bone marrow, or thymus to the peripheral secondary lymphoid organs. Now, this number two class, we need to first make an understanding of what's the difference between your immature, your mature, and the word naive. Now, whenever you would hear immature class, this would refer to the lymphocytes. This would refer to the lymphocytes that have not yet reached or that have not yet matured in the bone marrow or thymus. So please understand that class. Whenever we deal with your immature lymphocytes, these are lymphocytes that have not yet matured in the bone marrow or thymus. Now, if you still remember, if naalala nyo pa in the previous module, the bone marrow class, that this is the main organ for hematopoiesis. The bone marrow would create stem cells, and these stem cells would become lymphocytes. Now, your small lymphocytes specifically, you still remember yung small lymphocyte nyo. The small lymphocytes class would either stay in the bone marrow where they would become mature B cells. There or another one is that they would go to the thymus and they would mature to become T cells. So please take note of that. Now whenever you would call it a mature lymphocytes, these are the lymphocytes that have matured either in the bone marrow or the thymus. This is basically a mature B cell or a mature T cell. Well, the word naive class, please take note, is that these are T cell or B cell that have not been exposed, that have not been exposed to antigen. Basically, class, whenever you would hear the word naive T cell, naive B cell, they have not been exposed to antigen. Hindi pa sila nakaka-encounter ng bacteria, fungi, or viruses. They have no antigenic stimulation. Now, for you to understand that, class, for you to understand the entire explanation of this immature, mature, and naive, I'm going to show you something. So, imagine, class, this would be the bone marrow. Now, in the bone marrow class, there are two things that would happen whenever you would create, whenever we would deal with lymphocytes. The first one is that certain percentage of your small lymphocyte, the small lymphocyte would either stay in the bone marrow or they would travel to the thymus. Now, if they stay in the bone marrow class, the small lymphocytes would become B cells. If they travel to the thymus, they would now become T cells. Now, these B cells and T cells class are your mature small lymphocytes. So they could either be B cell or T cells. Now, your B cells class and T cells, both of this would now be released from the bone marrow. They would now be released into the circulation. Now, from the circulation class, they would now go to the secondary lymphoid organs, specifically class the lymph nodes and even the spleen. Now, if during this time while they are traveling in the circulation, please take note that they are considered naive T cells and B cells. Basically, they have not yet been exposed to antigenic components. Now, when they go to the secondary lymphoid organs, such as the lymph nodes and the spleen, their class, they would encounter antigens. They could encounter it in the lymph nodes or in the spleen, and from their class, they would become they would now become activated activated T cell or B cell. If you still remember, class, diba, in the lymph nodes, you have your primary follicle, your secondary follicle with the germinal centers for the B cell and so on. So that's basically the explanation, class. So just to repeat it again, the bone marrow class would usually be the site where small lymphocytes would mature. Now, the small lymphocytes class has two possible ways. They would either stay in the bone marrow or travel to the thymus. Now, if they stay in the bone marrow, they would become B cells. If they travel into the thymus, they would become T cells. In their primary organs, they would mature, becoming mature B cell, mature T cell. Now, they would now be released into the peripheral blood or the circulation, where they would be considered as naive T cell and B cell. Now, as they circulate into the peripheral blood, they would enter the the secondary lymphoid organs, such as the lymph node, the spleen. In their class, they can encounter an antigen, and that antigen could cause them or would make them become activated T cell and B cells. And if there is an activated T cell or B cell class, the T cell, just to add on, could either become your T helper or your T cytotoxic, while the B cell could become a plasma cell or a memory cell, if you still remember in the previous module. So that's basically your function number two. But we'll explain that further in the coming slides. So I hope that was clear. And then three, deliver of effector lymphocytes from the secondary lymphoid organs in which they were produced to sites of infection in any tissue where they would perform their protective function. Now here, class, there is the word effector. Now, whenever you would hear the word effector, this is somewhat synonymous to the word activate. The delivery of effector lymphocytes from the secondary lymphoid organs, as I mentioned earlier in the diagram I tried making. The secondary lymphoid organs, class, this is where they would be exposed to antigen. And again, if they are exposed to antigen, the naive T cells or B cells would become activated. So from the activated form, once they are activated, class, they would now be required to go to different sites of infection to perform their protective function. So let me repeat, the third main function of cellular migration is the delivery of your activated T cell, B cell from the lymph node, spleen to the site of infection to perform their protective function. So I hope that was clear. If you have questions, type them in the chat box. Now, leukocyte migration, class, is governed by the following principles. The first one is that naive lymphocytes would continuously migrate into secondary lymphoid organs, while lymphocytes that have previously been activated with antigen would migrate into tissues when there is no infection or tissue injury. Okay, it goes like this, class. As I mentioned earlier, if your mature T cell and your B cell, they would just, once they are released from the bone marrow, the thymus, they would go into the circulation, and from the circulation, class, they would go to the secondary lymphoid organs such as the lymph nodes and the spleen. Now, what happens here is that there are two possible ways. I mentioned one earlier. The first one is that it would encounter an antigen. If it encounters an antigen, class, it would become activated. And once it is activated, class, it would now go to the site of injury or site of infection. But whenever, let's say, there is no antigenic stimulation, there was no antigen found in the lymph node in the spleen, what happens is that the lymphocytes, secondary lymphocytes, will go back, will go out of the spleen, will go out of the lymph nodes, and will return back into the circulation. So let me repeat, class. Naive lymphocytes would continuously migrate mainly into the secondary lymphoid organs, while lymphocytes that have previously been activated with antigen would migrate into tissues where there is infection or tissue injury. So whenever your mature T cell and B cell are released from their bone marrow or thymus, they would go into the circulation, they would enter the lymph node or the spleen, and two things could occur. They could encounter antigen. If they encounter an antigen, they would become activated or would become an effector. And they would go to the site of injury or infection. But if there is no antigenic response, they would just continuously migrate into the secondary lymphoid organ. Meaning, class, they would just go back to the circulation. Another principal, class, governing migration is that adhesion of leukocytes to the endothelial lining would involve adhesion molecules found on the surface of both leukocytes and endothelial cells. Now, another thing you need to consider, class, is that there are certain receptors. The other word for receptors here, class, is the word ligands. Now, ligands or receptors could be found, class, in the walls of the blood vessel. Whenever you would hear the word walls of blood vessels, class, this would refer to the endothelial lining or the endothelium. So imagine if this is your blood vessel. This is where the blood would flow. On the surface class, there would be certain receptors or ligands. And that is where your WPCs would attach. Now, another thing to consider is that certain leukocytes also would have different um, adhesion molecules. So imagine this would be your sorry for the low pangit. What do I draw this? Would be your neutrophil. Example, let's say this would be your neutrophil. In the surface of your neutrophil, they would also have certain adhesion molecules, which we will tackle later, known as the selectins and your integrins. We will tackle them later. So these are the principles of your leukocyte migration. So I hope that was clear. Now, endothelial cells at sites of infection in tissue injury are activated by cytokines released by sentinel cells in the tissue, which would result to increased expression of adhesion molecules and chemokine. Okay, whenever you would hear class the word sentinel cells, this would usually refer to the macrophages. Now, macrophages class are typically found in the tissues, as you all know. Monocyte, if you still remember, they are found in the circulation. So if your monocytes go into tissues, they would become macrophages and they would have different names. An example, if it goes to the liver, they are known as your Kupffer cells and so on. Now, the sentinel cells class, whenever these sentinel cells would encounter an antigen, let's say it encountered an antigen, it would now release certain chemicals known as your cytokines. They would release cytokines class, allowing it to cause the proliferation or the activation of adhesion molecules, and chemokines. So let me repeat class. Endothelial cells at the site of infection and tissue injury are activated by cytokines secreted by sentinel cells in the tissue, resulting to increase adhesion molecules and chemokines. So when sentinel cells they encounter antigen, they would release cytokines, activating the endothelial cells, activating adhesion molecules, and even chemokines. Now, because microbes and necrotic tissue would induce the expression of the molecules that mediate the leukocyte endothelial adhesion, effector leukocytes would migrate to the endothelium mainly when and where they are needed. Now, yung microbes class and necrotic tissue, they are capable of creating molecules. Now, these molecules class are further known as your chemokines. This chemokines class would now mediate leukocyte endothelial adhesion, effector leukocytes, causing them to migrate. They will cause the migration of WBCs, the T cell and B cell, when and where they are needed, even the neutrophils and the monocytes. So I hope that was clear. Now, let's talk about your adhesion molecules. Now, adhesion of circulating leukocytes to endothelial cell is mediated by two classes of molecule, the selectins and the integrins. Now, selectins and integrins class have their ligands or their receptors. So we'll tackle them one by one. Let's start with your selectins. Now, selectins class, please take note, are plasma membrane carbohydrate binding adhesion molecules that mediate an initial step of low affinity adhesion of circulating leukocytes to endothelial cells lining the post capillary venule. Now, Selectins class are typically found, they are typically found in the surface of the endothelium. They are found in the surface of the endothelium class, and they would only provide a weak initial attachment or affinity. Later class, it will explain how they would become a stronger attraction or attachment. And they also, let me repeat, selectins, please remember, are found on the surface of the endothelium and would only provide an initial or weak affinity. Now, there are two main types of selectin class. The P-selectin, your CD62P, and the E-selectin or the CD62E. Now, let's tackle them one by one. The P-selectin was named P-selectin class because it was first found in platelets. The P here would stand for the word platelet and is stored in the cytoplasmic granules of endothelial cells and is rapidly redistributed to the luminal surface in response to histamine from mast cells and thrombin generated during blood coagulation. Okay, imagine class, this would be your endothelial lining. Now, inside here, class, there would be granules, cytoplasmic granules. Now, inside those cytoplasmic granules is where your P-selectin would be found. Now, whenever, let's say, this would be your macrophage class, let's say, would release cytokines. Once it releases cytokines class, these are the messengers, it would cause your P-selectin to be released, causing its ligands to appear on the surface of the endothelial wall. And this could be rapidly distributed due to histamine from mast cells and thrombin generated during blood coagulation. So if asked in your exam, how does P-selectin redistributes itself? The answer would be due to histamine from mast cell and thrombin during blood coagulation. So thrombin class is generally generated whenever you would have an injury or an opening. In your hematology, it would be discussed that your thrombin is the one responsible for converting fibrinogen into the fibrin. And the fibrin would act as a net to capture cell debris and so on. So I hope that was explained, class, what is PSEP. Now let's go to another type of selectin, your E-selectin. E-selectin was synthesized and expressed on the endothelial cell surface within 1 to 2 hours in response to the cytokines interleukin 1 and tumor necrosis factor. So please take note of that, class. E-selectin class is found, this is found in the endothelial cell surface. And they are usually produced due to the presence of interleukin 1 and tumor necrosis factor. So if asked in your quiz or exam, what causes the creation or the expression of your E-selectin, or which cytokine causes the expression of E-selectin, your answer would be interleukin 1 and tumor necrosis factor, which are all produced by your dendritic cell and macrophage in response to an infection. Microbial byproducts such as your lipopolysaccharide would also stimulate E-selectin expression on endothelial cells. So again, let's have another example, class. Imagine this would be your endothelial. Now, in your endothelial lining, there would be endothelial cells or the endothelium cells. Inside this endothelium cells, class, so imagine this would be the granules. This is where the P-selectin is. This is where you would find the E-selectin. Now, whenever your macrophage class would secrete cytokines such as interleukin 1 and tumor necrosis factor, it would cause class the expression of your E-selectin. So the E-selectin would cause its ligands to be released and so on. Now this could also occur, let's say there was a bacteria. Let's say one of the common Corynebacterium or the Bacillus corynebacterium would attach to the endothelial surface. When that happens, class, E-selectin could also be expressed. So please take note of that. Then a third selectin class, this is not part of the main selectins. We just stick with the P and the E selectin. The reason for that class is because this L-selectin, the third type, is found on leukocytes. So if you still remember, ang P-selectin nyo and E-selectin nyo, they are found in the surface of the blood vessel or the endothelium. But this L-selectin is different class. It is the only selectin found on leukocytes and not on endothelial cells. So if asked in your exam, which selectin is found on leukocytes, the answer would be your L-selectin, CD62L. Now, L-selectin on neutrophils would promote, so this is the function class, it would promote the adhesion of neutrophils to the endothelial cell. It would promote the adhesion of neutrophils to the endothelial cell that are activated by interleukin 1 and TNF and other inflammatory cytokines. Now, in adaptive immunity, L-selectin is required for naive T and B cells to home or to go into lymph nodes through specialized blood vessels called high endothelial venules. So this high endothelial venules class, just to give you an idea, this is the entryway of naive or inactivated T and B cell to the lymph nodes. And for that to occur, for them to enter this, the one that serves as a guide, is the L cell. So do take note of that. So these are the summary class. So we have the selectins, three types, the P and the E and the L. Now P selectin is also known as your CD60. Distribution, when you say distribution, where they are found class in the endothelium activated by histamine or thrombin, and the ligand, the molecule in the cell type. We have the sialyl Lewis X on PSGL1 and other glycoproteins. The cell type target, please take note. So this is the molecule class. Sialyl Lewis X on PSGL1, and the cell target is the neutrophil, the monocyte, and the T cells, effector and memory. For the E-selectin class, endothelium activated by cytokines, TNF1 and interleukin 1. The ligand here is the sialyl Lewis X. This is the name of the receptor class. Please take note of that. The example is CLA-1 on glycoprotein. You will find the meaning of this CLA class dyan sa module niya. So just look it up. And the target is the neutrophils, the monocyte, and the T cell. Then for the L-selectin, as mentioned earlier, they are found in WBCs such as neutrophil, monocyte, T cells, naive and central memory, and even B cells. The name of the ligand is the sialyl Lewis X or the PNAD on glycam1, CD34, and MADCAM1, and others. So the target would be the endothelium, specifically your HEV. So that's your solution. So we're done with the first group of adhesion molecules. Now let's go to the second one. The second one class is the integrins. Now the thing you need to remember about integrins is that they are found in cell surface, meaning they are found in the surface of your WBC. They mediate the adhesion of cells to other cells or to the extracellular matrix through specific binding interaction with various ligands. They increase their affinity for their ligands in response to intracellular signals which are induced in all leukocytes by chemokine binding to chemokine receptors, and in T cells by antigen binding to antigen receptor. Okay, for you to understand this class, let's say here you would have your selectins and their corresponding ligands. Now, on the surface of certain WBCs, such as neutrophils, you would find integrins. Now, what happens is that these integrins would roll and they would attach with their corresponding ligands. So, just for you to understand better. So, this would be the ligand for the selectin, and this would be the ligand for the integrin. Now, what happens, class, is that the ligand, as mentioned, would have a target. So, it would now attach to your neutrophil. Now, whenever your selectin would attach to your cell, this is a low affinity, as mentioned earlier. Now, here comes the important part, your integrin. Now, your integrin has yet to attach. So, imagine this would be the selectin again. I'll just enlarge the WBC. So, the selectin has already attached to its target cell, the WBC. Now, the integrin would now attach also to its corresponding ligand or target, to its corresponding target on the endothelium. When that happens, class, this will now form a stronger affinity. So let me repeat, class. Earlier, as mentioned, your E-selectins or your selectins as responsible for low affinity. What happens is that they would have a single attachment to their corresponding target cell. And when that happens, the integrins will now form another attachment to the corresponding target endothelium, forming a stronger affinity class. So that's the explanation. So just to give you another reminder again, whenever we talk about selectins, they are usually found in the endothelial surface. But for your integrins, they are found in the cell surface or the WBC surface. So please take note of that. So these are the integrins class. So we have your LFA-1, CD11A, CD18. They are found in neutrophils, monocytes, T cells, and B cells. And they would target ICAM1, CD54, ICAM2, found in the endothelium. Then we have your integrin, MAC1, CD11B, CD18. They're found in neutrophils, monocyte, and dendritic cell. Their target is the ICAM1, CD54, ICAM2, CD102, found in the endothelium. Then we have also the integrin VLA-4, CD49A, CD29, alpha 4 beta 7, CD49D, CD29. They are found in monocytes, T cells, monocyte T cells, the gut homing naive effector, and B cells, the gut homing. They would target your VCAM1, and found in the endothelium. VCAM1 and MADCAM1 endothelium in gut and gut associated lymphoid tissue. So that's your integrins. Now, another thing you need to take note of, class, is your chemokines. So chemokines will stimulate leukocyte movement and regulate the migration of leukocytes from the blood to the tissue. So basically, class, chemokines are the ones that would call WBC to the site of injury. Cytokines also would call them, but cytokines are mainly used, class, to activate the different adhesion molecules. So that's one way you can differentiate them. So, chemokines class is a large family of structurally homologous cytokines that stimulate leukocyte movement and regulate the migration of leukocytes from the blood to the tissue. Now, the chemokine is a contraction of the chemotactic cytokine. Okay, class. These are the different chemokines class, and they would have different functions. Now, they are subdivided into at least four groups. The first group class is the CC chemokines. They would include CCl2, CCl3, CCl4, 5, 11, 17, 19, 21, 22, 25, 27. All of them class have different or have an original name. And they would also have a target receptor. They have a major function class. So please, on your own, try to memorize this. This may be included in your coming exam. Alright, let's continue. So I won't tackle on this class to save time, but try to study them or memorize them on your own. Now, the chemokines class have different biological functions. Now, in inflammatory reactions, chemokines would serve to recruit circulating leukocyte from the blood vessel into extravascular site. Chemokine would play two roles of inflammation: increase adhesion of leukocytes to the endothelium, migration of leukocytes through blood vessels and toward the site of infection or tissue damage. Now, they are involved in the development of lymphoid organs, and they would regulate the traffic of lymphocytes and other leukocytes through different regions of the secondary lymphoid organ. They are required for the migration of dendritic cells from the site of infection into draining lymph nodes. Now, this is their main function. Increase adhesion of leukocytes to the endothelium and migration of leukocytes through blood vessels and toward the site of infection or tissue. Now, they also have certain steps class for the leukocyte endothelial interaction. The first one is the selectin-mediated rolling of leukocytes on the endothelium, chemokine-mediated increase in affinity of integrins, stable integrin-mediated arrest of leukocyte on the endothelium, and transmigration of leukocytes through the endothelium. So those are the steps. Now, for you to understand that, dito tayo class punta. Okay. Now, what happens here is that please take note that there are four things that would occur whenever there would be WBC endothelial interaction. So they would include rolling, integrin activation, stable adhesion, and migration through the endothelium. Now, all of this class will begin when your macrophages, whenever your macrophages would be stimulated by a microbe. Now, if it's stimulated by a microbe class, such as an antigen or a bacteria, a fungi, it would release your cytokines and it would also release your chemokines. Now, the cytokines, such as your tumor necrosis factor and interleukin 1, will activate certain selectins class. They would activate certain selectins, and when that happens, class, the WBCs would also arrive. Now, when the WBCs arrive, class, please remember that selectins would attach to their corresponding ligand or receptor. And these are usually found in their target cell. Now, since if you still remember, ang selectin, ang target niya is found on the surface of cell, what happens here, class, is that the selectin ligand, itong pink na to, it would now roll. Magro-roll yan, class, papunta sa cell surface kasi tinatawag siya ng mga receptors niya. Once they are attached or once they have rolled in the endothelial surface, hahanapin ngayon ni selectin yung ligand niya. And when that happens, class, it would form an attachment. But as mentioned earlier, this is a weak attachment. So what happens next is that it would now require your integrin. Now, integrin class is found in the surface of your WBC. Now, your integrin, the green one here class, will now look for its corresponding ligand. Nasaan ba yung ligand ng integrin nyo or receptor? The receptor of the ligand class is found in this in the endothelial cell surface. So what happens is that the integrin will now bind or will now attach to its corresponding ligand. And when that happens, class, it will now form a high affinity state, leading to the so-called stable adhesion. Now, one of the characteristics, class, of your WBC is that they are capable of squeezing. Kaya nilang mag-squeeze, class, through the tissues. And this action or ability is known as your diapedesis. Now, with the help of certain chemokines, lalabas yung mga chemokines, class, diba? Si chemokines nyo will guide them. So, using the chemokines, class, it would guide your WBC, and your WBC now will squeeze. Mag-i-squeeze yan, class, to the tissue or the site of injury. Now, once it is inside the tissue or the injury, it would now look for the target. It would now look for the target antigen, and that's how the WBC endothelial attachment would occur. So I hope it was understandable how the selectin, the integrin, and the chemokines help. Chemokines, class, as mentioned earlier, diba, they serve as a homing signal to find where the organisms are, the target antigen is. So let's continue. Now, migration of neutrophils and monocytes to tissue sites of injury or infection. So neutrophils and monocytes are usually found at the site of infection and injury. Neutrophils are the first leukocyte to migrate. Monocytes would follow after a few hours. Now let's talk about the migration of T cells. Yes. So lymphocytes are continuously moving through the blood, lymphatic vessels, secondary lymphoid organ, and non-lymphoid tissues. And distinct population of lymphocytes will show different trafficking patterns through this site. Okay. For you to understand this, class, let's go back again. So let's just focus on your T cell or your T lymphocyte. What happens here is that from the bone marrow, class, from the bone marrow, the small lymphocyte would go to the thymus, and in the thymus, class, they would mature and they would now become T cells. So we have here a mature but inactivated. Let me emphasize that, mature siya, but it is inactivated or naive. Now, the inactivated or naive T cell, class, would leave the blood and it would enter the lymph nodes through the high endothelial venule. As mentioned earlier, this is the entryway. This is the entryway of your lymphocytes, of your T cell and B cell. Now, the entryway would be guided by the L-selectin. So, punta tayo dito sa picture, class, para maintindihan. So, here you have your naive T cell. This is the inactivated. So, ito yung galing sa thymus, class. Pupunta lang sa circulation, paikot-ikot lang. It keeps on circulating. Now, once it enters the lymph nodes, it would enter, class, via the high endothelial venule. So, papasok siya dito sa HEV. Now, once it's inside the HEV, class, it would enter through the efferent lymphatic vessel. Then from the efferent lymphatic vessel, class, it would now enter the lymph nodes. Now, inside the lymph nodes, class, certain cells, such as your dendritic cells, your macrophages, they would now carry antigen, and they would also go to the lymph nodes, to the lymphatic vessel. Now, if your inactivated T cell, class, encounters, ito. This is your inactivated T cell. Ito yung dendritic cell or macrophage. Whenever your inactivated T cell would encounter or recognize an antigen, it would cause, ito yung antigen class. Di ba if naalala nyo yung sa phagocytosis, kukuha yung MHC molecule ng remaining molecules ng pathogen. Dadalhin nyo ng dendritic cell or ng macrophage. And from their class, pag na-expose tong inactivated T cell na to, it would now cause your inactivated or naive to become activated. And once they are activated class, they would now leave again through the efferent lymphatic vessel. Then they would go through the thoracic duct, which would enter now to the superior vena cava of the heart, and then ultimately into again arterial circulation. So they would leave class, they would go back to the circulation, and so on. Now, since they are already activated class, they are exposed to the antigen. As mentioned earlier, kapag activated ang mga T cells nyo and B cells nyo, they would typically go to the site of infection. So some would go to the circulation again, and some would go back. Some would directly go to the site of infection. So it would go to the peripheral blood vessel and finally into the site of injury. So I hope that was explained. Okay. Ulitin ko class para maano nyo. From the top. So from the bone marrow again class, your small lymphocyte will go to the thymus. In the thymus class, it would mature becoming a T cell. So you have your mature T cell, but this is considered inactivated or naive. Now, the naive or immature T cell would now go to your circulation. So punta tayo sa picture ulit, class. So ito yung immature T cell nyo or inactivated T cell. Now, the inactivated T cell or the naive T cell will now go to the lymph nodes. They would enter to the high endothelial venule. They would pass through the efferent lymphatic vessel and would enter the lymph nodes. Now, in the lymph nodes class, if there are certain cells such as your dendritic cell, macrophages that would carry the antigen molecule, and if they are exposed to, and they would expose it to the inactivated T cell, it would cause class your inactivated T cell to become activated. Now, once it's activated, it would leave the lymph vessel. Sorry. It would lead to the efferent lymphatic vessel, to the thoracic duct, emptying into the superior vena cava of the heart, leading again back to the arterial circulation, then going to the peripheral circulation. Now, since it is activated class, it would now go into the site of infection or inflammation. So I hope that was clear. So that's how your migration of T cells would occur. Just remember, class, how it passes through the lymph nodes. So the first entry is the high endothelial venule, efferent lymphatic vessel. It would also exit through the efferent lymphatic vessel, thoracic duct, then into the superior vena cava, then into the heart. Now let's go naman this time to your lymphocytes, your B cell. So before I explain this, let's read this. Naive B cells would use the same mechanism as naive T cells to home to the secondary lymphoid tissues throughout the body, which enhance their likelihood of responding to microbial antigen in different sites. Now, regression of B cells from secondary lymphoid organs would depend on S1P. Follicular B cells in the spleen would migrate to the marginal zone and then are carried by fluid to the red pulp and into the circulation. S1PR1 deficient follicular B cells have diminished ability to leave the spleen. Subsets of B cells committed to producing particular types of antibodies will migrate from secondary lymphoid organs into specific tissue where they differentiate into long-lived plasma cells. Okay, for you to understand this class, let's go back again. So balik tayo dun sa bone marrow. So the bone marrow class would release your small lymphocyte and they would stay in the bone marrow. Mag-stay siya sa bone marrow class. And their class, they would mature into become B cells. Now, this mature B cell is naive B cell. It would now go into the circulation. Now, compared with your T cell class, the B cells would tend to go more into the spleen. But it doesn't mean that it wouldn't go to the lymph nodes. So pupunta yan ng spleen and lymph nodes then class. Same with your T cell. Now, once they are in the spleen class, they would also enter, or once they are in the lymph nodes, they would also enter through the high endothelial venule. Now, inside the high endothelial venule class, they would now encounter, they would now encounter, they would now encounter antigen. Now, whenever they would encounter antigen, please take note that the naive B cell would go into the follicles. Now, in the follicles class, known as the primary follicle, if they encounter, let's say they encounter an antigen, it would cause the primary follicle to generate germinal centers. Once the germinal centers are created, it would transform the primary follicle to become your secondary follicle. And in the secondary follicle, the B cell will now become activated and it will now start creating blast cells, specifically the plasmablasts or become memory B cell. So ulitin ko class from the top. Your bone marrow would create or would secrete the small lymphocyte. It would stay in the bone marrow. And if it stays in the bone marrow class, if it stays in the bone marrow, it would now become a mature B cell. But this is inactivated or naive. Now, the mature B cell would either go to the spleen or the lymph node. So, punta muna tayo class sa lymph node. Now, sa lymph nodes class, pag dumating sila sa lymph nodes, before entering pala the lymph nodes, they have to pass through the high endothelial venule. Now, once they are inside the high endothelial venule, they would now enter the lymph node and they would go to the follicle, specifically the primary follicle. Now, if let's say there is an antigen present, if there's an antigen present, it would cause the creation of germinal centers. And this will cause your primary follicle to become a secondary follicle. Now, si secondary follicle nyo class will cause the B cell activation. Now, as you all know, as mentioned in the previous module, si B cell nyo would either become a memory B cell or a plasma cell. So if it becomes a plasma cell, it would now first become a plasmablast and or it could become a memory B cell. Now, your plasmablast class, please take note, have what you call different receptors or molecules. These are known as your S1P and your S1PR1. Now, please take note, class, that S1PR1 deficient B cells have diminished ability to leave the spleen. So kapag deficient, there is no S1PR1, the B cells would stay in the spleen. While your S1P allows it, class, to leave the secondary lymphoid organ. So please take note. So once it's a plasmablast, the plasmablast class will now go, excuse me, the plasmablast and memory B cell would exit from the lymph node and will go back into the blood. Now, here's what happens. There are two possible things that would happen. The plasma blast, if it's an IgG plasma blast, it will migrate into the bone marrow. Once it's in the bone marrow, it will now become an IgG secreting plasma cell. If it's an IgA plasmablast, it would now go to the mucosal surfaces, becoming IgA secreting plasma cells. So if you take a look at this class, these are the chemokines when they are introduced to the chemokine and so on. So I hope that was clear. So let's go for another rundown for the third time para maintindihan niyo pa. Again, for the third time, the bone marrow class would secrete your small lymphocyte. The small lymphocyte would stay in the bone marrow and it would now become a mature B cell. The mature B cell class is sadly naive or inactivated. Now, once that happens, class, they would go to the circulation, they would either go to the spleen or the lymph node. So let's focus on the lymph node. So it would enter the lymph node class via the high endothelial venule. So pumasok na siya dito class sa high endothelial venule. It would now migrate into the follicles class. So mapupunta siya sa tinatawag nating primary follicle. Now once it encounters an antigen via certain chemokines and so on, it encounters antigen class, it would now cause the creation of your germinal centers. The germinal centers class will cause it to be converted into secondary follicles. And in the secondary follicles is where the B cell would now become activated. The activated B cell would either become a plasmablast or a memory B cell. Now, please take note, class, that in the plasmablast, it can secrete certain substances, such as your S1P and S1PR1. Now, please take note that S1PR1 deficient B cells have weak ability to leave the spleen. So once that happens, the plasmablast would and memory B cell would exit the lymph node. And they would go to the blood. And from the blood class, two things would happen. They would either become the IgG plasmablasts migrating into the bone marrow or the IgA plasmablasts migrating into the mucosa. If it migrates into the bone marrow, it would now become your IgG secreting plasma cell. IgG class is a class of antibody. We will tackle that further in the module 6. While if it's an IgA plasmablast, it would go to the mucosa, becoming IgA secreting plasma cell. IgA class is also another class of antibodies. So I hope that was clear. Let's go to your acute inflammation class. So accumulation of leukocytes, plasma proteins, and fluid derived from the blood at an extravascular tissue site of infection or injury. So there are four cardinal sites, redness, swelling, heat, and pain. So rubor, edema, calor, and dolor, and yung loss of function, yung fusiolacia. Major events in the process of inflammation would include increased blood supply to the infected area. This is typically caused class by vasodilation. In your HEMA 2, I believe it was discussed that in your primary hemostasis class, whenever there would be an injury or inflammation, there would be increased blood flow via vasodilation. Then increased capillary permeability caused by retraction of endothelial cells lining the blood vessel. So the capillary walls or the blood vessel walls would become permeable. They would become easily passed through. Then migration of white blood cell, mainly neutrophils, from the capillary to the surrounding tissue, and migration of macrophages to the injured area. Acute phase reactants would stimulate phagocytosis of microorganisms. Then other substances involved would be your histamine secreted by mast cells. This is the one responsible for the vasodilation of the blood vessel, bringing additional blood flow resulting to redness and even heat. Acute phase reactant would initiate and control the response. Neutrophils would migrate 30 to 60 minutes after injury. Major cell type in acute inflammation. Neutrophil immigration would last 24 to 48 hours. Then macrophages would migrate to the site of injury several hours later and would peak 16 to 48 hours. Attempts to clear the area through phagocytosis. Then this is how basically class the summary of how your WBC would move to the site of infection. So resident macrophages and mast cells would release chemokine, causing vasodilation and inducing selectins would loosely bind circulating leukocytes. So ito yung tinatawag ko kanina class na weak affinity, causing them to roll. Now, chemokine-induced integrins class would cause a stronger affinity. Integrins enable the leukocytes to crawl between the endothelial cell via diapedesis. Then leukocytes will then follow the chemokine signals to the site of infection, known as your chemotaxis. Then we have your chronic inflammation, a process that takes over from acute inflammation if the infection is not eliminated or if the tissue is prolonged. Involves recruitment and activation of monocytes and lymphocytes. It can go to undergoing tissue remodeling with angiogenesis. So when we talk about angiogenesis, this is the creation of new blood vessels and fibrosis. So that basically ends your module. So these are the references class. If you have any questions, type them in the chat box or raise your hand. Thank you.